Yes, very good evening to all of my students. Yes, really I am back. I was, for some reasons, for some medical issues, I was not present over here. Chilam ne ghanta. I was in Chennai for some procedure for my parents. Okay, let us start with the topic that we have. We need to start, and that is about meiosis. We are going to start with meiosis topic. I think I have given you some idea about meiosis. The first stage of the meiosis called prophase 1 of meiosis. Especially we have talked about the 5 substages of the prophase 1. I think I have already talked about leptotin, zygotin, diplotin, diakinesis. Leptotin, zygotin, achitin, diplotin, and diakinesis. These 5 stages we have already talked about in the very last class, if you remember. But today I'm going to just give you some idea, and we are going to complete this particular topic. See, meiosis. What is meiosis is all about? We have to understand first. Meiosis ta kake bole? What is meiosis? Ye kya hai? Basically, what is meiosis? Meiosis is, normally we know that for a life to be formed, we need the union of 2 gametes. We need the union of 2 gametes. So, for example, say this is sperm with chromosome number N, and this is ovum, and this is having chromosome number N. Now this is ovum, which is a result of what? Is a result of meiosis, because the body from which the ovum has been produced, this body is having diploid number of chromosomes. This body is having diploid number of chromosomes. Fine. Now the entire body is diploid, so only in her ovary this process called oogenesis takes place. And during oogenesis, what happens? The ovum is being produced. And by the process of oogenesis, which is possible by the help of meiosis 1, and then we need the help of meiosis 2. We need the help of meiosis 2. So I think the basic idea you have already got. Which one is the real meiosis? Which one is the real meiosis or reductional division? It is the meiosis 1, which is called as the reductional division. It is the meiosis 1, which is called as the reductional division. That means what happens from one particular cell, which is having 2n number of chromosomes, which is called as the primary oocyte. It undergoes meiosis 1. It undergoes meiosis 1 and forms into 2 cells, each having half the number of chromosomes. So the parent cell was having 2n diploid number of chromosomes, and by the process of meiosis 1, it results into 2 cells, each having half the number of chromosomes, and that is NN. And these 2 cells are secondary oocyte cells. Secondary oocyte cells. Okay. Now see, these again, the secondary oocyte cells again undergoes meiosis 2. And this one is called as a polar body. We will talk about that. Fine. So ultimately what is going to happen is ovum is going to be produced by the process of meiosis 1 and meiosis 2, both. Now meiosis 2 is as good as mitosis. Meiosis 2 kya hai? Meiosis 2 exactly mitosis ki tarah equational division hai. Okay. Now, so this is ovum being produced. How is sperm being produced? Sperm being produced by the process of, very good, spermatogenesis. And this takes place where? Inside testis, you know that. And again, if this is the first cell having 2n number of chromosomes, tell me what is the cell called? The cell is called as primary spermatocyte. Primary spermatocyte. And this primary spermatocyte undergoes what? Undergoes meiosis 1 first. It results into formation of 2 cells. These 2 cells are now called as secondary spermatocyte. Secondary spermatocyte. And this secondary spermatocyte undergoes meiosis 2, meiosis Roman 2, and forms into these. From this NN, they are going to, by the process of meiosis 2, which is called mitosis, is going to result into formation of the same number of chromosomes, that is N and N, and this cell is now called as spermatid. From each spermatid, by the process of differentiation or spermiogenesis, it forms into the sperm or spermatozoa. So there are 4 numbers of spermatozoa produced from the primary spermatocyte. So ultimately, what is the significance of meiosis? The chromosome number reduces. It helps in the decrease in the chromosome number one. Number two, it helps in variation. It helps in variation. Without meiosis process, variation is never possible. Clear? So there are two aspects or two significance of meiosis. So ultimately this M and N, this is one cell, this is another cell. The union between two cells is what is this called? The union between two cells is called syngamy. It's called syngamy. And ultimately it's going to form what? Zygote. It's going to form zygote. Now, so when we talk about meiosis 1, so this meiosis 1 is going to have stages like this. What are the stages? The first one is prophase 1. The second stage is metaphase 1. The third stage is anaphase 1. And last one is telophase. Telophase 1. And what about meiosis 2? The same one. Meiosis 2 will be is going to consist of the 4 stages, and those are prophase 2, metaphase 2, anaphase 2, telophase 2. Fine. Now, if I make the cell cycle of meiosis, tell me what are the stages? What are the stages? Now you tell me what is this? This is prophase 2, metaphase 2, anaphase 2, telophase 2. What is this? G1, S, G2, prophase 1, metaphase 1, anaphase 1, telophase 1. What is this? What is the stage? This is the cell cycle for meiosis. And this gap between meiosis 1 and meiosis 2 is interkinesis. The gap between meiosis 1 and meiosis 2, this is meiosis 1, and this is from this to this is meiosis 2. And this is called interkinesis. It's very, very important. But mind you one thing, what happens over here, the difference between the mitosis and meiosis is basically the replication of DNA never takes place over here. Replication of DNA is not needed because already replication occurs in the meiosis 1. During meiosis 2, there is no replication of DNA because it's not needed. Clear? It's very, very important. Now, see, in prophase 1 of meiosis 1, what are the stages? The stages are, क्या stage हैं? 5 substages हैं. There are 5 substages are present. 5 substages. What are those? The first one is leptotene, then what? Zygotene, then pakaitin, diplotene and diakinesis. Is this clear? Now these are the 5 substages, and mind you, whenever examiner wants to ask questions, the favorite question for them is this stage. And if you remember in the last class we have talked about leptotene. Teen means what? Thread. Teen means thread. Lepto means what? Thin. Leptos means thin. So thin thread. So what is a thin thread? What we consider about the thin thread? The chromatin materials, they remain just like a thin thread. The moment they are going to make into zygo means zygos, teen means thread. So what they are going to show? They are going to show a complex called synaptonymal complex. So mind you, this synaptonymal complex is a very, very important concept. Synaptonymal complex. And they are going to make what? They are going to make, tell me the term. Tetrad. What is the other term for that? Bivalent. What is the difference between tetrad or bivalent? Are the same? Yes, they are same. How? Yes. How many chromatids? Four chromatids. Four chromatids. But chromosomes are two of them. Two of them are the chromosomes. So two chromosomes, but how many chromatids? Four. So as because they are 1, 2, 3, and 4 chromatids, we call it tetrad. But how many chromosomes? Two. So it's called bivalent. It's called bivalent. So this occurs due to the formation of complex called synaptonymal complex. In packaging, which is very, very important, what happens? These two are sister chromatids of each other. These two again are sister chromatids of each other. But these two together, these two are non-sister chromatids of the homologous chromosomes. So what happens in packaging? What happens in packaging is, in packaging, what happens? There's a recombination occurs. There's a recombination module which occurs. Recombination module which occurs. And this recombination between these two, so there's an exchange of the genetic material between these two. And what happens? What happens is the process is called as crossing over. So crossing over takes place where? Starts in packaging stage. And the enzyme which is responsible for this is recombinase. The enzyme which is responsible for this is recombinase. You can just visit the earlier video. We have talked about that. So we need to go a little bit faster. Diplotin. What happens in diplotin? What happens? The place where they are going to join. So there is a thread-like structure. They make a nodule-like structure, recombination nodule. And ultimately, what is going to happen in diplotin stage? The crossing over gets completed in diplotin stage. It starts in the packaging. And the crossing over completes in the diplotin stage. What is the stage called? Synapse. What do we call it as? The place where the exchange takes place, there is a formation of X separate structure. What is that called? The X separate structure is called chiasmata. X separate structure is called chiasmata. Now in diplotin, the crossing over completes. But what happens in diakinesis? Terminalization, you can see in the book, terminalization occurs in diakinesis. That means when they are going to connect one particular place, and slowly they are going to reach, they're going to start separating with each other. The moment they start separating with each other, that is the point when they are going to show as terminalization. So in diakinesis, what is most important is terminalization takes place. Is this clear? So prophase 1 is very, very important with the aspect that the main crossing over takes place over here. This is unique only in case of meiosis. But we haven't seen this the same process occurring in mitosis. Got the point? So this is very, very important about prophase 1. What happens after that? What happens after that? After the exchange has been occurred, what is going to happen? Metaphase 2. Metaphase 1. So in metaphase, what is going to happen? We know in metaphase, what is going to happen? We know the term is called equatorial plate. Remember? Equatorial plate. So in equatorial plate, this is the centriole, and this is the centriole, and this is the aster formation, which ultimately forms into spindle fibers, and the chromosomes, they are going to remain in where? In the equatorial plate. They are going to remain in the, arrange themselves in the equatorial plate. So this is the equatorial plate. But what we can see some segments are present which are different, which are different. What has happened over here? Some exchange has occurred. And this exchange has been due to what? This exchange has been due to what? Due to crossing over. Okay, due to crossing over. So this you can see in your NCERT the same. If you see the NCERT about this stage, what it says, see, we come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. So ultimately what happens? In the gamete formation, during the gamete formation, it helps in what? It helps in meiosis takes place, which helps in the formation of haploid gametes. The key features of meiosis are as follows. What is that? The key features. What is that? Meiosis involves two sequential events, two sequential cycles of nuclear and cell division called meiosis 1 and meiosis 2. So there are two sequential events. One is meiosis I and the one meiosis II. But only a single cycle of de-replication. De-replication takes place only once, not twice. That is very, very important. There's a single cycle of de-replication. In meiosis II, there never needs any de-replication. That's what I have said to you that now. Okay, this is 2007. The question came in 2007. Now you see, meiosis I. So meiosis I contains prophase I, metaphase I, anaphase I, telophase I, meiosis II, prophase II, metaphase II, anaphase II, telophase II. Now, the most important, meiosis I is initiated. Mind you, how to study NCERT? All these points are very, very important. These points are very, very important. Each point is going to be one option for you. Meiosis I is initiated after the parental chromosomes have replicated to produce identical sister chromatids at the S phase. At the S phase. Meiosis involves pairing of homologous chromosomes and recombination between, what is it? Non-sister chromatids of homologous chromosomes. What is non-sister? These are non-sister. These are non-sister. Say for example, this is another one. Okay, and the exchange is going to take place between these two. So these two are what? These two are non-sister chromatids of homologous chromosomes. 4 haploid cells are formed at the end of meiosis II. So at the end of meiosis II, how many haploid cells are going to get produced? 4 haploid cells. And for that purpose, for that understanding, we need to understand this one first about spermatogenesis. Get this idea. Spermatogenesis. Spermatogenesis. This takes place where? Inside testis. Spermatogenesis in human takes place inside what? Inside testis. And inside testis, if this is the scrotum, the two testes, this is a canal which is called as inguinal canal. The testis slowly descends down inside the scrotum. Because the scrotum contains, and this happens when during the gestation period, when the male baby is inside the mother's womb and the age of the male baby is close to 210 days, that means 7 months old. During that time, on an average, the testis which was initially present inside the abdomen, it slowly descends down inside the scrotum. And this process is very, very important process. Why? Because inside the scrotum, there is a type of muscle called as dartos muscles. And these dartos muscles, by contraction and relaxation, can reduce the temperature of the testis by minimum 2 to 3 degrees Celsius, which is required for spermatogenesis. So what is the normal body temperature of human being? 37 degrees Celsius. But for spermatogenesis test, for the process of spermatogenesis, it needs close to 34 to 35 degrees Celsius. And this is provided only by the scrotum. And that is why there's an extra abdominal sac which comes out of the body in majority of male mammals. Got the point? In order to make what?
To reduce body temperature.
To reduce body temperature, to reduce the temperature of the testis so that it can help in spermatogenesis. So this is spermatogenesis. This cell is called primary spermatocyte. This is primary spermatocyte. And it undergoes what? It undergoes meiosis I first. It undergoes meiosis I first, which results in the formation of 2 cells, each having half the number of chromosomes, and that is why meiosis I is basically called as reductional division. It is called as reductional division. So these 2 cells are now called as secondary spermatocyte. These 2 cells are now called as secondary spermatocyte. Now tell me what is going to happen with these 2? Meiosis II is going to take place. Very good. Meiosis II is going to take place. Meiosis II is going to take place. And this meiosis II is basically called as mitosis. Forms into 2 cells, each having NN and each having N and N. These cells are called spermatid. These cells are called spermatid. Fine. And then it's going to form into what? It's going to form into spermatozoa. What is the process? This step is called differentiation. Very good. So this cell has been differentiated to form a tadpole-like structure called spermatozoa. But there is a technical term for this. This stage, if this is spermatozoa, what is this stage called? This process is called spermiogenesis. Spermiogenesis, or it is also called as spermateliosis. Is this clear? Any issues with this? Come on. Now, in your book, you can see a diagram of this cell. There is a first cell where it forms in two cells. The first cell you can consider it to be the primary spermatocyte. You can write it down with a pencil. It's the primary spermatocyte. Then it forms two cells. You can write it down. It is a secondary spermatocyte. Then in the next page, you can see 4 cells are present, which is called as spermatid. And these spermatid ultimately forms into what? The sperms. Got the point? Now, but this whole process takes place by the help of 2 main steps, meiosis I and meiosis II, and that is sequential. What is the meaning of sequential? One after another. So meiosis I is going to take place first, then followed by meiosis II. Clear? This is very, very important. Now, let's get back to this meiosis I. Prophase of the first meiotic division. Prophase of the first meiotic division is typically longer. Prophase is typically longer and more complex when compared to the prophase of mitosis. Why it is more complex? Agnishwar, why it is more complex? Because it is having how many stages? 5 substages. What are the name of the stages? Leptotin, zygotin, pachytin, diplotin, and diakinesis. It has been further subdivided into the following 5 phases. Okay, we just talked about this. Okay, diplotin and diakinesis. This is leptotin. The first step is what? Leptotin. Just take it out. Lepto means what again? Let's repeat it. Lepto means what? Thin. Thin means thread. So it's a thin thread. During leptotin stage, the chromosomes become gradually visible under light microscope because it's completely thin. So under light microscope, it's visible. But see, the compaction of chromosomes continues throughout leptotin. What is the meaning of compaction? Condensation. This is followed by the second stage of prophase I called zygotin. So zygotin. What happens in zygotin? Zygote matlab kya hai? Zygote means n plus n equals zygote. That means pairing of something, and for pairing, what is required? Some complex is required. What is the complex? Synaptonymal complex. Very, very important. During this stage, chromosomes start pairing together, and this process of association is called synapsis. Such paired chromosomes are called homologous chromosomes. क्या कहते हैं उसको? These paired chromosomes are called. See how many years this question came. 2010, 22. Electron micrographs of this stage indicate that chromosomes, synapses, is accompanied by the formation of a complex structure called synaptonymal complex. Star mark. So whenever you get to see synaptonymal complex, what should come immediately in your mind? We are talking about zygotin stage. Rita, is this clear? Good. Now, the complex formed by a pair of synapsed homologous chromosomes is called a bivalent or a tetrad. Bivalent or a tetrad. So how many chromosomes? There are 2 chromosomes, but how many chromatid? 1, 2, 3, 4. And that is why these 4 together is called tetrad, and 2 chromosomes makes it bivalent. Visible at the next stage. Vishal, is this clear? Okay. The first 2 stages of prophase I are relatively short-lived. Which one? Leptotin and zygotin. Short-lived. As a whole, it is a longer one. The first 2 short-lived, but the next 3 compared to the next stage, that is pachytin. The most important step is what? Pachytin. Because what happens in pachytin? Tell me a single word. Crossing over. Or recombination. Recombination takes place. Okay, pachytin. During this stage, the 4 chromatids of each bivalent chromosomes becomes distinct and clearly appears as tetrads. अब distinct होकर क्या बन गया? Tetrad. And you can see this through light microscope only. No issues with this now. Okay. This stage is characterized by the appearance of recombinant. See, nodules, recombination nodules. So this term is very important. Nodules term is very important. So recombination nodules. That is fine. The sites at which crossing over occurs between non-sister chromatids of homologous chromosomes. Crossing over is the exchange of genetic material, that is genes, between the 2 homologous chromosomes. See how many years the question came. So what is this? Crossing over takes place between what? 2 non-sister chromatids of homologous chromosomes. 2 non-sister chromatids of homologous chromosomes. Crossing over is also an enzyme-mediated process. And the enzyme is called recombinase. What is the name of the enzyme? Recombinase is the name of the enzyme. Star point. So recombination में कौन help करता है? Recombinase enzyme help करता है. Crossing over leads to recombination of genetic material on the two chromosomes. The recombination between homologous chromosomes is completed by the end of Pachytene. So recombination gets completed at the end of Pachytene, leaving the chromosomes linked at the sites of crossing over. So it remains in the form of what? Linked. It remains in the form of linked in the sites of crossing over. The sites of crossing over. So they remain in the form of linked, called crossing over. And what is the point where they get linked? It makes an X-shaped structure called chiasmata. So what is the next stage? Diplotin. So what happens in diplotin? The beginning of diplotin is recognized by the dissolution of, what is the meaning of this? Dissolution of the cyanonyl complex. Now it's going to get dissolved. Dissolution of the cyanonyl complex. So what is going to happen? They're going to get separated now. And the tendency of the recombinant homologous chromosomes of the bivalents to separate from each other except at the sites of crossing over. So they are going to join, but only what is going to happen? They are going to come over, they are going to get dissolution only in the sites of crossing over. These X-shaped structures are called chiasmata. Very, very important. The X-shaped structure are called chiasmata in the year 2020 in oocytes of, very, very important, in oocytes of some vertebrates. The diplotin can last for months or years, and that is why in some of the oocytes of vertebrates, they remain in the arrested phase in this prophase 1 of this stage. Prophase 1 of meiosis I, they remain in the arrested phase. And it can take place it can continue with this arrested phase for some months or for some years in the oocytes of some vertebrates. Is this clear? Any issues, Babu? Great. Now, this diplotin, and what is the last one? Diakinesis. The final stage of meiotic division. Diakinesis is what? It is the final stage of meiotic prophase 1. Meiotic prophase 1 is diakinesis. This is marked by, what is the term? Very good. So tell me one word for each steps. Leptotin. Leptotin. Thin thread. Thin chromatin threads. Next, zygotin. One word. Cyanonyl complex. Pachytene. One word. Recombination. Crossing over. Enzyme. Recombinase. Diplotin. Chiasmata. And what happens? Crossing over completely takes place. It gets completed. Diakinesis, one word. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosomes for separation. So they get ready for separation. By the end of diakinesis, the nucleolus disappears. Tell me, Varun, in prophase of mitosis, when we talk about prophase of mitosis, what happens? The nuclear membrane completely gets disappeared. The nuclear membrane starts disappearing. Nucleolus starts disappearing. Endoplasmic reticulum, Golgi body, they start disappearing. And what? The centriole starts moving to the opposite poles. Remember, the same thing occurs over here. Nucleus disappears and the nuclear envelope also breaks down. Diakinesis represents transition to the metaphase. Diakinesis आ जाना मतलब किस में चले जाना? कौन से स्टेज में चले जाना? Prophase completed and now you are stepping into metaphase 1. Clear? Any issues with this? So what is the specialization in prophase 1? Crossing over this is the specialization. Now metaphase 1 of meiosis and metaphase of mitosis are all the same. अब देखो क्या है? Metaphase. The bivalent chromosomes align on the equatorial plate. है ना? याद है अभी बोल रहा था आपको equatorial plate में अगर ये cell है, if this is a cell, so if this is a centriole, fine spindle fibers, and the chromosomes, they are going to align themselves in the equatorial plate. So they are going to align themselves in the equatorial plate. Okay. The microtubules from the opposite poles of the spindle attach to the pair of homologous chromosomes. Who will tell me what is the site where spindle fibers are going to get attached to? Kinetochore. Very good. Kinetochore. Fine. If there is one particular chromosome if there's one particular chromosome, how many kinetochore will be there? For one particular chromosome, how many kinetochore? 2. Very good. Mind you that. Fine. And mind you, this is in case of mitosis. This is in case of mitosis. But right now, in meiosis II, in the stage of metaphase 2, what we want to see, this is a chromosome. इसको याद रखना है, ये क्रोमोसोम है, ठीक है? एक क्रोमोसोम के साथ एक kinetochore, एक क्रोमोसोम के साथ एक kinetochore. That's the difference. This we are going to see in metaphase 2 of meiosis II. meiosis, ये डिफरेंस देखोगे आप. माइटोसिस में क्या था? यहां पे एक kinetochore था, यहां पे एक kinetochore था with one chromosome, यहां पे 2 क्रोमोसोम के साथ, ठीक है? Okay. Anaphase 1, the homologous chromosomes separate while sister chromatids remain associated at the centromeres. Anaphase में क्या होता है? It's going to pull to both the poles. Fine. Now, at anaphase 1 stage of meiosis, the genetic constitution of gametes is finally decided. अब यहाँ पे anaphase stage में the genetic constitution of the gametes is completely decided in the anaphase stage. आना means किस तरफ आना? पोल की तरफ आना. आना किस तरफ? पोल की तरफ. And that is called anaphase. Okay. Now, telophase. The nuclear membrane and nucleolus. बोलो क्या होगा telophase में? What happens in telophase? Reappears. So it reappears. Cytokinesis follows, and this is called as dyad of cells. है ना? कितने सारे सेल्स? 2 ना? एक सेल से 2 सेल बन गया. अब 2 सेल में क्या होगा? meiosis II. क्या होगा? meiosis II. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus. The stage between the 2 meiotic divisions is called as interkinesis. Between meiosis I and meiosis II, there is a gap, and that gap is called interkinesis. The stage between the 2 meiotic divisions is interkinesis, and it's generally short-lived. It's very, very short-lived. There is no replication of DNA. देखो, no replication of DNA during interkinesis. No replication of DNA. It's a very, very important one. There's no replication of DNA during interkinesis. Star point. Clear? Star point. अब देखो ये diagram आपके book में है. This is the diagram. Prophase 1, and then you can see some other structures over here. Book में diagram में देखना थोड़ा सा other structure. मतलब Prophase 1 में क्या हो गया? What has happened? Crossing over has taken place. Some part of the genetic material has been exchanged. So you can see some discoloration in some of the part, and ultimately prophase 1, metaphase 1, anaphase 1, and telophase 1, it forms into dyad of cells. Now this 2 undergoes meiosis II. तो एक सेल से कितने सेल बनेंगे? 2. और इस सेल से कितना बनेगा? 2. Having chromosome number NN. That's why it is called as equational division or mitosis. So meiosis II is as good as ये 2 सेल था. देखो prophase 2 बना, prophase 2 से anaphase 2. फाइन. पर याद रहे anaphase 2 में यहाँ पे इन prophase 2 में यहाँ पे duplication of the centrioles is going to occur. 4 centriole बनेंगे, 2 यहाँ पे आएगा, 2 यहाँ पे आएगा. Fine. And ultimately form into this telophase 2. In telophase 2, what we can see is 4 cells, each having n chromosome number. Any issues with this? This particular process we have seen in spermatogenesis. Okay. Now meiosis II. Meiosis II is initiated immediately after cytokinesis, usually before the chromosomes have been fully elongated. In contrast to meiosis I, meiosis II resembles a normal mitosis. है ना? Prophase 2 में क्या होगा? Interkinesis followed by prophase 2, a much simpler prophase than prophase 1. क्योंकि यहाँ पे क्या नहीं है? What is not there? Agnishwar? No such sub-stages. No such sub-stages. Because prophase 1 में 5 sub-stages थे. The nuclear membrane disappears by the end of prophase 2, as usual. यही तो काम है ना इसका. Nuclear membrane starts disappearing. The chromosomes again become compact, and now they're going to start compacting, because compaction के बाद ही क्या होगा? metaphase 2 में आएगा वो. At this stage, the chromosomes align at the equator, and microtubules from opposite poles of the spindle get attached to the kinetochores of sister chromatids. Anaphase 2 में क्या होगा? It begins with the simultaneous splitting of centromere of each chromosome, which was holding the sister chromatids together, allowing them to move towards opposite poles. So ये दोनों को खींचेगा opposite pole पे. of the cell by shortening of microtubules attached to the cytopore. Microtubules jitna shorten hoga, it is going to go towards the pole, fine, attached to the cytopore. Meiosis II is the equational division due to the separation of chromatids. Telophase II. Meiosis ends with the telophase II, in which the two groups of chromosomes once again get enclosed by a nuclear membrane. अब क्या होगा जब division हो जाएगा, they are going to start forming nuclear membrane again. Cytokinesis followed, resulting in the formation of tetrad of cells, that is 4 haploid daughter cells. कितने सारे haploid हो गए? How many daughter cells you are getting? 4 of them, each having n number of chromosomes, that is called haploid. A somatic cell that has just completed the S phase of its cell cycle, as compared to gamete of the same species, has same number of chromosomes and twice the amount of DNA. Very, very important. यहाँ पे देखो. What is important? A somatic cell that has just completed the S phase of its cell cycle, as compared to the gamete of the same species, has same number of chromosomes. Chromosome कितना रहेगा? N. But twice the amount of DNA. Amount of DNA is not going to come down. Twice the amount of DNA. Amount of the DNA will remain the same, that is 2N, 2C. Okay? So the chromosome number is going to become half, but the amount of DNA will remain the same. What is the significance of meiosis? Significance of meiosis is what? Say, कहाँ पे लिखा है देखो, decrease in the chromosome number. In meiosis, the daughter cells differs from parent cell as well as amongst themselves due to the segregation of, it's called independent assortment. क्या कहते हैं इसको? Independent assortment and crossing over. I think you have heard about independent assortment where? In genetics. What is the law? Mendelian, which law is independent assortment? Third law. Law of independent assortment. Meiosis is the mechanism by which conservation of specific chromosomes number of each species is achieved across generations in sexually reproducing organisms. Even though the process per se paradoxically results in the reduction of chromosomes number by half. Reduction in the chromosome number by half. Okay, it also increases the genetic variability in the population of organisms from one generation to the next. See, genetic variability. The second one is what? Genetic variability. First point is what? Reduction of chromosome. Second point, genetic variability. Variations are very important for the process of evolution. So without variation, you cannot have evolution. So what is the one word we can use? What is the significance of meiosis? It is by the help of meiosis only evolution is possible. Clear? Evolution is possible only by the help of meiosis only. Reorganization of genetic material or genetic recombination occurs during meiosis. देखो, reorganization of genetic material is possible only in case of meiosis. Meiosis is evolutionarily significant because it results in the recombinations. And that is why none of us will look alike except some monozygotic twins. But even in monozygotic twins, there are some variations in the form of texture of the hair. We will talk about that, resulted by polygenic inheritance. That's all about meiosis. If you have got any doubt, you can ask me. Otherwise, this is all about meiosis. Now, mind you, if I ask you some of the questions, if you try to answer it, if I ask you, what is the major difference between mitosis and meiosis? What is going to be the major answer? What should be the answer? What is the major difference between mitosis and meiosis?
Mitosis does not help in radiation.
Mitosis only helps in increase in what? Increase in cell number and also increase in the cell growth. Cell size increases and the cell number increases, cell size increases. Do you remember nucleocytoplasmic ratio? If it is increased, then the chances of mitosis is more or less more. Nucleocytoplasmic ratio. If you have missed that video, just have a look. Fine. In the very first class about mitosis, we have talked about why mitosis occurs. If the nucleocytoplasmic ratio increases, it results into mitosis. Now, mitosis and meiosis, the difference is in mitosis, the chromosome number remains the same and it helps in the growth. It helps in mainly growth. And without growth of an organism, the organism cannot reproduce. The organism cannot reproduce. So what is important? First, mitosis is required, and then meiosis is required to have life. So meiosis is what is the importance of meiosis? It is only by the meiosis that the chromosome number reduces to half. The chromosome number reduces to half. And this chromosome number, when it reduces to half, it first takes place by the help of meiosis I, which is basically called as reductional division, and then these cells undergoes meiosis II, which is also called as mitosis or equational division, which results into formation of the same number of chromosomes is going to come over here, forming what? N number of chromosomes. And that N number of chromosomes, which is there in the gametes, is going to unite with the other gamete, male gamete or female gamete, and ultimately form into a new life, forming zygote. And there will be intermingling of the genetic material between the two nucleus, which we are going to call it as amphimixis. And after amphimixis, what is amphimixis? Intermingling of the genetic compositions, intermingling of the genetic constitutions, and ultimately it will form into variations, and that leads into different identity, different individuals. Is this clear? So what is the take that we can get it from this particular chapter? The first take, significance of mitosis. What are the main importance of mitosis? Why mitosis occurs in a cell? What are the parameters which are important for a cell to undergo mitosis? These are most important from mitosis topic. When we talk about meiosis, prophase 1 comes immediately in our mind. All the substages of prophase 1 is very, very important. What is the stage where the cyanotomical complex is going to form? What is the stage where recombinase enzyme is going to come into play? What is the stage where terminalization is going to occur? What is the stage where dissolution of this chromosome is going to occur? We are going to talk about all dissolution of cyanotomical complex is going to occur. So these are most important one. And then comes the importance of the significance of meiosis. Otherwise, all are same. And mind you, nuclear membrane disappears, nucleus disappears, and when it comes back, fine, sister chromatid, non-sister chromatid of homologous chromosomes. Crossing over takes place between two non-sister chromatids of homologous chromosomes. Very, very important. Otherwise, this chapter has got nothing, but you have to go line by line of this chapter. I'm going to make a short video on the importance of the chapter, what are those chapters which you are going to study line by line of NCERT, where much more information is not needed. Extra information is not that needed. And what are the chapters where you need to have some extra informations? I'm going to make a video, just have a look. That's all for today. We have completed this chapter called cell, cycle and cell division. So next day we're going to start with a new topic. Clear? Start solving questions. Whatever question bank you have got, you can solve those questions, or you can take the questions from me. Okay, that's it. Chalo, that's it.